Around a half century ago, no one can imagine that you can manipulate or engineer an organism to perform a predetermined function. In the 1970s, the first recombinant DNA technology were produced using restriction enzymes and introduced to host organism, which is a bacterial cells, to produce a desired traits or functional traits, which start a whole new and extended area of researches. My name is Yusuf Mohamed, and I will talk about computational design and analysis of recombinant DNA using in synthetic biology. The computational biology and bioinformatics in recombinant DNA techniques. This computational workflow of recombinant DNA technology describe how we can discuss the role of computational algorithms and tools in the designing and analysis the recombinant DNA. Firstly, we will talk about vector design. For a program or an algorithm to design a good vector, it must take into consideration the four most important parameters. First, origin of replication. The origin of replication on the vector must fit with the host enzymes. Secondly, selectable marker. The vector must include a gene that can be selected for using external stresses that is not normally expressed in the host organism. Third, the vector size. The vector size must be compatible with the host organism. Finally, the host compatibility. The vector must be compatible with the plasmid in the host organism. This table shows the most important and recent computational tools using in the design vector. Discuss gene design. New computational tools have been developed recently to help scientists generate their own synthetic gene and structure instead of optimizing design for biological activity. The gene design software aims to facilitate the redesign of protein coding genes based on predefined properties of interest, like the parameter of vector design. Gene design software must consider the five most important parameters that affect gene expression. Firstly, codon use bias. Second, codon context bias, and RNA secondary structure, and ribosomal binding site, and finally restriction site and the hidden stop codon. The table shows the most recent computational tools used to gene design. After creating the recombinant DNA sequence with the target gene in it, an order is made with gene script to synthesize the whole sequence. Use a synthesizer for 0.19 per base, which might sound like a lot of money for the whole sequence, but it will probably less than what you would pay with the restriction enzymes method due to the large number of errors. Step is prediction of best method of transformation. After design the recombinant DNA, we have to transfer this recombinant DNA into an organism or the host cell. So the next step is transformation process, which occur in the wet lab. This step cannot be designed using dry lab techniques, but we can use them to predict the best method for transformation given in specific target organism or species before we begin this process. For example, there are 6 strains of E. coli and 4 different methods of transformation shown in the figure. In a study, we can determine the best method for a specific strain. The statistical tests used for this are ANOVA and t-test. After the recombinant DNA is introduced into the target cells, the real-time PCR and RNA sequencing technique help us qualitatively analyze the amount of mRNA in the transformed cells. After the recombinant DNA is introduced into the target cells, real-time PCR and RNA sequencing technique help us quantitatively analyze the amount of mRNA in the transformed cells. The results of these techniques are analyzed using algorithms to differentiate between mRNAs before and after transformation. The figure showing that the real-time PCR analysis workflow أو benchmark. This figure show bioinformatic tool typically used for RNA sequencing data analysis. What all of this indicates is that dry lab techniques are going to become a big part of the field of synthetic biology, and that in the near future most of research in this area is going to be done on computers rather than in wet lab. This is the references, and thank you. What all of this indicates is that dry lab techniques are going to become a big part of the field of synthetic biology, and that in the near future most of research in this area is going to be done on computers rather than in wet lab. This is the references, and thank you.